Hello everyone, welcome to Practically's NEET course, and today we are going to talk a little bit about the biological classification. So in the previous class I was discussing a little bit about the things such as the hierarchy and the taxonomy, and we were discussing a little bit about the levels of taxonomy, the phylum, the kingdom, the class, families, etc. So that it is easy to differentiate between the animals so that we can understand the characteristics of the animals. But now what we have to do is we know that now that there are around 1.7 to 1.8 million organisms in the world, okay, different species. Now, not all of them are similar. There are higher organisms such as humans, other animals. There are plants. There are lower organisms such as bacteria and amoeba, etc. So many things are there. We cannot put everything in one block, right? We have to segregate them. So for that reason, we want to go for the biological classification. Okay. So first, I'll talk a little bit about Practically. Now, as you all know, that Practically is India's first experiential app, which runs for the grades 6 to 12th. We have a lot of courses. We have a lot of coding classes. We have regular courses. We have robotic classes. So many things are there. You can just download our app and check it out, and I'm sure you'll feel that it is one hell of an experience. It will be. Okay. So you can also follow us on Facebook, on Instagram, and like us on the YouTube. And you can rate your experience from your iPhones and your Android phones. Okay. So let's talk a little bit about the classification. Now, just now I told you that classification is what? Classifying. Classifying as in dividing the things. There's the division of things. Okay. We have, as I told you, let me just write some things and it will be easier for you to understand. Okay, so as you see, we have 1.7 to 1.8 million species in the world, which includes plants, animals, bacteria, etc., everything. Okay, so we cannot put everything in one box. Impossible. Okay, different characteristics, different type of growth, different type of simulations, everything is different. So just to make it convenient, okay, we have divided them, classified them into various segregations. Okay, so now what is biological classification, you might ask. Okay, now there are a few characteristics such as growth. Let me write it. There is growth, there is metabolism, there is reproduction. Okay, so many things are there. Okay, and for each and every aspect, okay, is different for each and every organism. So we cannot put it in one box, right? Impossible to put it in one box. So on basis of these similarities, and some organisms may have similar kind of growth, similar kind of metabolism, similar kind of reproduction, put them in one box. Different, then you put them in the other box. So these things are known as classification. Okay, I hope you understood. Let me just check. Okay, so anything you want to tell me, you can tell me on the chat box. Okay, and I can see your, if you have any doubts or anything, I can clear it out. Okay, that is how it works. Now let's move on. So now there are different kinds of classification. Okay, there is artificial system, there is natural system, and there is phylogenetic system. So these are just the different types of classifications. But after this, I'll just say a little bit of things about the different type of system, and then we'll jump on to the what we are doing right now, who started it and how it has developed. over the years. Okay, so the artificial system was first given by this gentleman. His name is Aristotle. Okay, and he was there around 3000 to 3000 years ago, in around 384 to 322 BC. BC is always written backwards, as you know. Okay, that is history. I won't go there anyway. So 384 to 322 BC. So what he saw at time, science was not very, and this gentleman, fellow gentleman, was what to say, he was a philosopher. At the time, science was not so developed. We did not have so many systems and internet, and that nothing was available at that time. It was just books. People used to write their thoughts in it, people used to read, and that's it. Okay, so although the things were a bit slow, but this gentleman, he was a very intelligent person, and he actually saw that there are some, I can find something, there are some animals which give birth through eggs. Okay, there are some animals which give birth through the body, and then there is egg laying, and so many things. So we can divide them. Okay, that's all he said. That's all he could say, because it was very long ago. Okay, so it was not possible for him to actually go through go through all the classifications. Now, his disciple is this man called Theophrastus. He was his disciple, and he actually saw that there are some plants which are small. Okay, these are grass and all, these are herbs. Okay, then there is shrubs. Okay, small middle sized plants, and then there are trees, huge hunks of trees like banyan, etc. So he divided them, that there are 3 to 4 kinds of plants. That's all he could say about that. Okay, so gone with the artificial systems. Now then there came natural system of classification. Okay, this was just based on the vegetative and floral characters of plants. They did not go into the animal kingdom a lot. These gentlemen, George Bentham and Joseph Dalton Hooker, they talked a little bit about the cryptograms and the phanerograms. Okay, these are cryptograms are which does not have seeds, no seeds, and no flowers. We'll talk about it in the next class. Okay, kingdom. Okay, I'll tell to you later. Anyway, so no seeds, no flowers. These are algae, bryophytes. Okay, and then there is phanerogams, which had seeds and flowers, proper reproduction and everything. These are normal like mango, coconut, etc. Okay, so they talk about the natural system of classification. What they did not consider here was the Darwinian theory, which was the evolution and all. They did not consider. So there was a discrepancy in this kind of method. Okay. Then there came the phylogenetic system. Phylogenetic system is what? Phylogenetic system is just dividing it into sections. Okay, tree kind of a thing. From plants can be divided into bacteria and embryophyta. Then came from their embryophyta, there came gymnosperm and angiosperm. See, you can see it over here. Gymnosperm and angiosperm. From angiosperm again monocot, dicot. From dicot again Archaeomaceti and Metaclamidaceae. Okay, so like that, the division. Okay, like this. That came in the phylogenetic system. That's all. That was also known as cladistics. Now here comes the main person who is also known as the father of taxonomy. This gentleman is named as Carolus Linnaeus. Okay, so he gave an idea about the whole process, the process of dividing the organisms into kingdom Plantae and kingdom Animalia. Okay, That's all he did. He did it in the year 1858. That is the 18th century. All he could do, this much only. Okay. He just divided into Plantae and Animalia. It included bacteria, mycoplasma, fungi, and photosynthetic plants. And then Animalia is unicellular and multicellular animals. What he did not actually consider fungi. He put it directly into the plant kingdom. He did not consider fungi. Okay, that is where it became a bit problematic and people started developing his idea. Okay, then from there, since as I said, he did not consider fungi and all, there is this person known as Ernst Haeckel. This was in the 19th century. Okay, in the 19th century, he developed from Carolus Linnaeus's theory that there are three kingdoms actually. There is Plantae and Animalia, as Carolus Linnaeus told, and another thing he included, that is kingdom Protista. Okay, in Protista, he included unicellular and colonial eukaryotes such as bacteria, algae, fungi, and protozoans. So in Protista only, he put bacteria, algae, fungi, and protozoa. Everything. Okay, this is actually wrong because it was developed later, but he put everything together in Protista. All the lower beings, I'll write it here. All the lower beings was put in the kingdom Protista. Okay. So now came the second last one. This was done by a person named Copeland. Okay. What he did, he put it in 4 different things: Monera, Protista, Plantae, Animalia. Okay. Then again, there was a drawback where fungi again was put in again in Plantae. Okay, he did not consider fungi. For a different fungi was considered as plant, which is not true because they have different kind of characteristics, right? Still, they put it in Plantae, which was wrong. So to sum it up, to do everything, okay, there were so many. I'll go back. Okay, it started with Aristotle. Okay, then came Theophrastus. Then there were they were talking about plants and not animals, and then they made the stepwise tree kind of a thing, which was in the phylogenetic system. And then came the main person in the world, the father of taxonomy, Carolus Linnaeus. He put just 2. Then Ernst Haeckel put 3. Copeland put 4. Now finally, there was a person called R.H. Whittaker who actually put the 5 system classification. Okay, so let me see if you have any questions, if there are any questions or anything. Okay, if there are any doubts, let me see. As of now, there are no doubts. Okay. If there are any doubts, please let me know. Anyway, moving on to the 5 system classifications. What happened? It was done. He took everything into consideration. All the primitive theories, Aristotle's theories, Carolus Linnaeus's theories, every theories he took into consideration. Okay. Then he based it on different sections. Okay. So what he did, see. Now, complexity of cell structure, there is a prokaryote. There are prokaryotes which are primitive in nature. This is primitive. Okay. And eukaryotes, these are non-primitive. Okay. Then there is complexity of organisms, body, unicellular or multicellular. It can be one cell. Okay. Be it prokaryote or eukaryote, mostly eukaryotes. Can be unicellular, it can be one cell, or it can be multicellular also. Okay. Then there is mode of nutrition. Okay. There is autotrophic or heterotrophic. Now, autotrophic is for the living beings which make their own food. Okay, and heterotrophic is for organisms which don't make their own food. Okay, so now major ecological role there are again apart from nutrition and complexity near the eukaryotes, prokaryotes and everything, apart from that they put major ecological role. Some are producers like the plants, some are consumers like the humans, and then there are decomposers. Okay, decomposers are fungi, molds, etc. Okay, and then there is the phylogenetic relationship, one after the other, how we are related to the primates. Okay, how cats are related to the tigers and the leopards. Okay, so accordingly he gave these 5 kingdoms. Okay, there is Monera, there is Protista, there is fungi which is also known as mycota, there is metaphyta or plantae, there is metazoa or animalia. Okay, so today we are basically concentrating on these 3. Okay, Monera, Protista and fungi. Plantae and animalia we will be discussing in the subsequent sessions. Okay, so first we'll start with Monera. Okay, now Monera is basically the most primitive kind of cells, the organisms which contains the most primitive kind of cells. Okay, the bacteria, the different kinds of bacteria totally is Monera. Monera is basically given to the bacterial species. Okay, now these are, as you know, unicellular or filamentous. These, see, as you can see here, they are filamentous. Okay, and they're omnipresent, they're present everywhere. You know, you can actually do this experiment. You take some kind of agar which can produce which can be some kind of nutrition, and you just hold it anywhere in your bedroom, or if you want to go to a hospital, like don't go right now, wear a mask and everything. But if you want to go outside, okay, best thing is you go outside and you just hold it in the air for some time. Okay, and then keep it. I'm talking about agar, okay, solidified agar. Hold it in the air for some time, keep it, and you keep it in your room for some time. You will definitely find out a lot of cultures within maybe 24 to 48 hours. You will see a lot of white dots in your Petri plate. Okay, that is why, because it is omnipresent, bacteria are present everywhere. It is also true for fungi also, but bacteria mainly. Okay, and different kinds of species you can find out. Okay, and then moving on, you can actually, there is cell wall, okay, which is composed of polysaccharides and amino acids, peptidoglycans or murine. Okay, different bacteria, some have peptidoglycans, some have murine. Okay, we'll discuss about it later. Then they are autotrophic, as in photosynthetic. Okay, they make their own food. And heterotrophic, some they don't make their own food, they totally depend on the decaying matter. Saprophytic or parasitic. Parasites as in they take a host like human body or some other kind of animals, etc. Saprophytic, this is dead animal or plants. Parasites can be any living animals or plants. Okay, and they reproduce via vegetative way and asexual methods. Mostly it is vegetative or asexual. There is no sexual reproduction seen in Monera. Okay, so as you can see, Monera is actually divided into Archaeobacteria and Eubacteria. Just write it over here. Monera is divided into Archaeobacteria and Eubacteria. Okay, so now Archaeobacteria and Eubacteria, why this division? Because Archaeobacteria is one of the most, there is definitely some like differences. That's why they were divided into two parts. Now, Archaeobacteria, why Archaeobacteria? They were archae as in like archaeological, as in historical. They were one of the first living things which were found in the on the earth, and they have a different kind of cell structure about which we'll be learning after this. Okay, different kind of cell structure. Their cell wall is a bit different, and they live anywhere. They can live in the harshest of conditions they can live in, okay, which is not possible for Eubacteria. Eubacteria can live in normal conditions, normal air, normal soil, normal water, okay, but Archaeobacteria can live anywhere. Because of their cell structure, they can actually tolerate the harshest of conditions. That is why it's known as Archaeobacteria. So now you can see there are some, these are the differences, okay, these are the differences here also, okay, these are the differences and these are the similarities. Sorry. Okay, so Archaeobacteria, they're often, as I said, they're often found in the extreme conditions, okay, below the ocean bed, okay, below it, okay, just at the bottom of the ocean you can find it. And apart from that, you can find it in hot springs, which is very hot. The temperature rises up to 100 degrees Celsius to 104 degrees Celsius, okay. Okay, and then the cell wall is not composed of peptidoglycan. It is composed of something called pseudopeptidoglycan. Okay, and there are some, these are some examples. There is Halobacterium and there is Thermoproteus. Okay, then Eubacteria, as I said, they are found in most of the non-extreme environments. Okay, and their cell wall is also composed of peptidoglycan, not pseudopeptidoglycan, peptidoglycan, and they are in different shapes. Although it is true for Archaeobacteria also, they can be of some different shapes. And examples are basic microbacteria such as Mycobacteria, Cyanobacteria, Nosta, Nabina, etc. Okay, so there are also some similarities, as you can see. Unicellular, this is given because it falls in Monera. Okay, no membrane bound nucleus, mitochondria, ribosomes. When you talk about bacteria, that time only you can actually say that there are no organelles. Okay, there are no organelles. Okay, there is no, apart from ribosomes, there is no mitochondria. The nucleus is just floating around in the cytoplasm. There's no nuclear membrane or anything. Nothing is there. Okay, so it's like that. And then there is reproduction by binary fission. This is asexual or vegetative. There is no sexual reproduction over here. Okay, so there are, again, let me just check if there are any. Okay, if there are any doubts, please let us know in the chat box. Okay, so as we go on with the types of Archaeobacteria, now we'll talk a little bit about the Archaeobacteria. Archaeobacteria is basically of three kinds. There is methanogens. Methanogens, basically, that is very easy to remember about because of their similar kind of name. So methanogens, because they convert CO2 of swampy areas into methane. Okay, this conversion, okay, carbon dioxide into methane, that is why it is called methanogen. The example is Methanosarcina barkeri. This is actually a picture of Methanosarcina barkeri. Okay, halophiles, these are salt loving. Any kind of salt pans, you can go and check any kind of salt pans, if somebody is mining salt somewhere there, for sure you'll find these kind of bacteria. One of them is Halobacterium salinarium. Okay, any saline in the Dead Sea, Salt Lake, Great Salt Lake, everywhere you can find this kind of bacteria, which is high in salt. Okay, and then there is the thermoacidophiles. These are the most harshest conditions they live in. Okay, like sea temperature approx 80 degrees Celsius to 100 degrees Celsius, hot springs and all, they can be found out. Some of them are Ferroplasma acidophilium. Okay, they like that. They use the acids and all, they use it for nutrition. Okay, so that is why they are called like that. And there is Pyrodictium abyssea. In the abyss of ocean, these kind of bacteria are found out. Okay, now we go to Eubacteria. Okay, There are actually 3 to 4 kinds of Eubacteria. Okay. Now, first is the Cyanobacteria, which have chlorophyll. That is one of the main things. They have chlorophyll. Okay, similar to green plants. They are unicellular, such as spirulina, and there is Anabina and Oscillatoria. Okay. Then there is chemosynthetic autotrophic. These make their own food. Cyanobacteria is actually autotrophic again. Okay. They make their, with the help of, they are totally photosynthetic in nature. Okay, chemosynthetic autotrophic, they are non-photosynthetic. They cannot go do photosynthesis, but what they do is they use this chemo thing, this chemicals, this radiations, etc. that is used for their nutrition. See, energy instead of light, they use chemical energy. Okay, they take chemical energy from somewhere. I'll tell you this. At the bottom of the ocean, okay, where you'll find a lot of chemical reactions happening because of the lava which goes around, passes around under our ocean and everything. So these kind of bacteria, they take that kind of chemical reaction, make their nutrient, make their food, and they use it up for their reproduction and other purposes, growth, reproduction, etc. Okay. Then there is heterotrophic bacteria. These are the most common kinds of bacteria. Any kind of heterotrophic bacteria, okay, they are abundant in nature. They're always there. Okay, lot of them of their species are there in the nature. It has significant impact on human affairs. They are helpful. Okay, helpful in making curd. Okay, antibiotics, fixing nitrogen. These is rhizobium. Okay, rhizobium helps in nitrogen fixation, making curd. Okay, then there is antibiotics. So many kind of bacteria, okay, are helpful for human beings. Okay, then there are some pathogenic also, like Vibrio cholerae. It causes cholera. Okay, Streptococcus, that causes E. coli. E. coli. E. coli causes digestive issues. Okay, salmonellosis. Okay, so many things are there which causes harm also, and it is helpful also for the human beings. Then there is mycoplasma. Mycoplasma is not actually comes in bacteria, but we had to put it somewhere, so it comes under. It's not real bacteria. Because of what? Because it completely lacks a cell wall. Okay, cell wall is one thing which actually makes a bacteria. Okay, so it completely lacks a cell wall. That's why it's not actually a real bacteria. These are smallest living cells known and can survive without oxygen, and many mycoplasma are pathogenic in animals and plants. Okay, so they are actually very pathogenic in nature, and they can live anywhere without oxygen. That is why it's pathogenic in nature. They can live anywhere inside our body without even needing of oxygen or anything, and they're not really bacteria. Okay, done. Okay, this is just, I wanted to put it here so that you can understand different kinds of Archaeobacteria and different kinds of Eubacteria, Archaeobacteria. See, thermoacidophiles, methanogenia, methanospirillum, etc. Then there is Eubacteria, there's Anabina, Nostoc, there are gram positives, gram negatives, etc. So we are done with Monera. Let's move on to Protista. Okay, so Protista is actually unicellular eukaryotes. These are small ones. Okay, unicellular, one cell. These have one cell. Okay, first let me check if there are any doubts for Monera. Bhuvan, if you want to ask something, what do you want me to repeat? Whatever you want me to repeat, you can just write it and I'll repeat it again for sure. Okay, what do you want me to repeat again? Please let me know. Okay, so now we'll go on to Kingdom Protista. If you want me to ask something, if you want me to repeat something again, I'll go back for sure. Until and unless I'm here, I'll just write what you want me to repeat and I'll do it. Okay, so we'll go to Kingdom Protista. So these are unicellular eukaryotes. These are one cell. Unicellular means one cell, just one cell. Okay, it will look like this, somewhat like this. These structures are Protistas. Okay, so now they are freshwater and marine and parasitic forms. Basically, they are they can be free living also. They are free living also, and they are parasiting also. Free living, also parasitic. Okay, so now cell wall is usually absent, and if present, it's impregnated with silica. Okay, mostly they don't have cell wall. Okay, and if there are cell wall, they have silica in that. That is a special feature, something called diatoms. They'll have a special feature where cell wall is basically silica. Okay, so that is a special feature, and you should remember that this comes a lot, this thing about diatoms with silica. Okay, this is a very important question for NEET. Okay, it has come a lot of times. Okay, so they are photosynthetic or non-photosynthetic. Okay, and their locomotory structure may be cilia, flagella, pseudopodia, or absent. Cilia, flagella, as you can see, these structures, this is flagella, then there can be cilia. Okay, and pseudopodia, this is actually a false feet kind of a structure. Okay, and they reproduce via sexual and asexual methods. This is unlike Monera, they have both sexual and asexual methods. So now Protista is actually divided into 4 or 5 kinds. Okay, there is Chrysophytes, Dinoflagellates, Protozoans, slime molds, and Euglenoids. I'm just saying the name right now. We'll try and understand what they actually are. So there are 5 types: Chrysophytes, Dinoflagellates, Euglenoids, slime molds, and Protozoa. Okay. So let me check. Okay. No. So first we'll start with Chrysophytes. These are actually algae kind. Okay, they are not actually algae. Algae has been put into a different kind of kingdom, plant kingdom, but these are algae type. Okay, and they are mostly found in fresh water, and they are photosynthetic in nature. They make their own food. Okay, then there is dinoflagellates. Have you ever seen bioluminescence? Okay, bioluminescence is actually a type of nature kind of thing which happens in the nature, where if you go at night into specific kind of beaches, okay, and in those beaches you can actually find that blue color things. You can see fluorescent blue color planktons, you can see. Okay, it will be shine, it will look like as if the stars have come down into the beach. It will look very beautiful. That is caused by this kind of structure called dinoflagellates. Okay, you can actually Google it and see how beautiful it is. I have seen it with my own eyes. Okay, and it is beautiful. Okay, bioluminescence. Okay, where these things, they'll come into your, in just like it will look like bluey in the water, bluish fluorescent. Okay, and these dinoflagellates, they are single celled, as you know. Protistas are single celled in nature, single celled eukaryotes, and mostly marine plankton. Okay, in freshwater habitats or beaches, etc. See, as you know, see bioluminescence, you can see. Some of the examples are Physteria piscicida, Noctiluca scintillans, etc. Okay. Then comes euglenoids. Euglenoids are actually photosynthetic in nature. Okay, as you can see in other kind of protistas also, they are photosynthetic in nature. And one of the most unique, this is also very important for NEET, is euglena. Okay, euglenoids have a covering called pellicle, which is a protein-rich membrane. Since they don't have cell wall, they have a protein-rich membrane, which is, this is just for euglenoids. It's known as pellicle. Okay, remember this, Pellicle, pellicle. Remember this. Okay, and they tend to supplement photosynthesis with absorption of organic compounds from smaller beings such as bacteria. Okay, sometimes when they are not photosynthesizing, they can get their nutrition from smaller beings such as bacteria. Okay, when there is absence, suppose there is no sunlight and they cannot photosynthesize. Okay, at that time they go for this, just eat up bacteria and take them as nutrition. Okay, so these are euglenoids. Examples are Euglenoviridis, Euglenogracilis. Okay, then there is slime molds. Okay, slime molds are basically sac-like structure. Okay, I'll write it. Sac-like structure. Okay, several kinds of unrelated eukaryotic organisms that live freely. They live freely. Okay, freely as single cells, but can aggregate together to form multicellular. Suppose they are seeing some kind of feature where there is no food. Okay, they are dependent on other organisms. So these slime molds. Okay, 1 second. Suppose there are many slime molds and there is no food. So what they'll do? They'll come together, become one sac-like structure, and then try to feed on something. Okay, so they share their nutrition. Okay, so one of the examples is Physarum polycephalum. This is also known as a blob. Okay, this one is also known as blob. Okay, protozoa, one of the main important examples of Protista. Let me check if there are any questions or anything. Okay, no. So we'll move on. So this is the main example of Protista. So these are protozoas, again single-celled organisms. Okay, they come in different shapes and ranges from amoeba. Okay, one of the main examples of protozoa is amoeba, which can change its shape to paramecium with its fixed shape and complex structure. Amoebas look very unique. Okay, they look somewhat like this. Okay, there's no shape or anything. Okay, so they can change their shape. Amoebas change shape. Okay, and because of the presence of paramecium. Okay, and they live in a wide variety of moist habitats, etc. And they have many life cycles. Some of them are infectious. Okay, one part of the life, when they go into a host or something, that time they are infectious. And they mostly reproduce by asexual means. Okay, there's no sexual reproduction in protozoa. Mostly fission, longitudinal fission, as in what it will be. Suppose this is an amoeba. How it will reproduce? It will form, suppose it formed a bud, and then this bud will be taken off. Suppose this is a bud, something. Okay, and then it separates. This is how they reproduce. Okay, so there's no sexual reproduction. Okay, this is how they reproduce. Now then there are the other things such as flagellates. Okay, these are parasites or free living. They have flagella for locomotion, that is why the name. They have this flagellates. Okay, they have contractile vacuole. Okay, which helps in the nutrition. Okay, there is trypanosoma, trypanosomes, giardia, leishmania. These are all different kinds of protozoas. Okay. Then there's amoeba, as I said, one of the most important things. They move by pseudopodia. Pseudopodia as in a false leg. Okay, they create their capture. Okay, suppose I'll show you now. Suppose this is an amoeba. They have a false leg, and this false leg keeps moving. Okay, so that the organism keeps moving. It goes round and round, round and round and round again, so that they can go and capture their food. Okay, so there's no definite shape, as I said, and there is no pellicle. Pellicle is just for euglenoids, nothing else. Pellicle is mostly for euglenoids. So the example is amoeba, entamoeba, etc. Okay. Then there is sporozoans. Sporozoans, you have heard about it because of plasmodium. Okay, this one causes malaria. Okay, plasmodium species. Okay, so this causes malaria. Okay, they are endoparasitic. Okay, they don't have any specialized organ for locomotion. Pellicle is present, which is subpellicular. They just go. Okay, their whole life cycle, most of the important part of life cycle is inside the host. Okay, and finally there is ciliates, which are aquatic and move actively with the help of thousands of cilia. As you can see, these things, these materials, they are known as cilia. They have a fixed shape, like unlike the amoeba and everything, due to covering of pellicle. Okay, this have ciliates and sporozoans, they have pellicle. Apart from euglenoids, we also have pellicle. Okay, some species have organ for defense called trichocysts. This is also very important for NEET. Remember, ciliates have defense organ. Okay, which is very important for NEET. They also move with the help of cilia. That is why the name. Movement of cilia also helps taking food inside the gullet. Examples are Paramecium, Vorticella, Palentidium. Okay, so most important part in ciliate is the trichocysts for defense. Okay, remember this part. Okay, you can find these PDFs, whatever I'm teaching, you can find these PDFs in our app. All you have to do is subscribe, and you can find everything there in the app. Okay, so anything else? Let's move on. Okay, the final thing which we are going to learn today is kingdom fungi. We have learned about Monera, we have learned about Protista. Now we'll learn a little bit about fungi. Now fungi, we are reading about this thing, we'll give a little bit more importance because of their reproducing capability. Okay, this becomes a little bit higher than Monera and Protista because of their reproducing capability. Okay, so first we'll see what it is, and I'll tell you a little bit about the diagram I put. Okay, so that it will be easy for you to understand. Now they are unicellular or multicellular. Okay, some are parasites, they are dependent on some of the organisms, and there are saprophytes which grow on dead leaves or dead animals or anything. Symbionts, where there is a give and take kind of a policy. Okay, and then there is where they produce their own kind of food also. The thalloid part is known as mycelium. Okay, so now how it looks is, this is the mycelium. Okay, so what they have, hyphae. They have something called hyphae. Okay, these are filamentous in nature. You know, fungus are filamentous in nature. Then together they have so much hyphae. This altogether is known as mycelium. Okay, and after so much filamentous, they grow into this kind of shape. Okay, this kind of an umbrella kind of a shape. Okay, now mycelium, as I said, is tubular thread-like structures, filamental structures made up of hyphae. And one of the main things, which is today's point, is it is the cell wall has chitin. Okay, apart from other things also, it has chitin, which makes it a different kingdom. Okay, just because it has chitin, it is a different kind of kingdom. Okay, now as I said, we are going to concentrate a little bit about on the reproduction part because of the different kind of reproduction stages it goes through. There's vegetative, budding, fission, etc. There's asexual, where there is use of spores, and there is sexual, where they use the spores and then they have sexual reproduction. These things, as you can see, these dot things, these are the spores. Okay, this is a very important part in the body of a fungus. Okay, let's move on and we know. Okay, so the first kind of they are actually divided into a lot of different kinds of things. Okay, but the first today we'll talk about 4 most important kinds of fungi. Okay, the first kind is Phycomycitis. Okay, now they are mostly found in aquatic habitats and on decaying wood, in moist and damp places, obligate parasites on animals. Okay, they are mostly dependent. They can be independent, but they are mostly dependent. Now the mycelium is aseptate and cenocytic. What this means? Aseptate as in, suppose mycelium, as I said, is filamentous, group of filamentous things. Okay, so this is how it looks. Okay, and there is no division, it is one long cell. Okay, that is why, okay, had it been like this, had there been divisions, this is septate. Okay, but this is aseptate. Because there are no divisions. Okay, and inside that only there are a number of nucleus. Okay, in septates also there are nucleus like this. Aseptate also has a lot of nucleus, that is why it's known as cenocytic. Okay, so asexual reproduction takes place by zoospores. These are the spores. Okay, motile or aplanospores. When they move, okay, when the spores are moving, they become zoospores. When they are not moving, they are stagnant, then they are aplanospores. Okay, these spores are endogenously produced in the sporangium. As you can see, these flowery like structures you can see, this is known as the sporangium, and from here only the spores go out. Okay, and Oomycetes, Zygomycetes are types of Phycomycitis. These are just types, okay, such as Phytophthora infestans, Rhizospus, Phytophthora infestis. It's actually potato blight disease. Okay, just there's a disease in potato plant just caused by Phytophthora infestans, and Rhizospus is normal fungus you find. You keep a damp bread in a dark room. Okay, that is where bread fungus is a Rhizopus. Okay, so this is Phycomycitis. Now we move on to Ascomycetis. Ascomycetis is basically, again, there is a sac-like structure. As you can see in this picture, okay, it's sac-like structure. Okay, and they are saprophytic. Saprophytic, as I have already said it, they are dependent on the dead animals or dead plants. Decomposers, of course, if they are saprophytic, they have to be decomposers. Parasitic or coprophilous, growing on dung. They can grow on dung also. Okay, Sexual spores are ascospores, which are produced endogenously, sac, like asci. Some common examples are yeast, penicillium, aspergillus. Okay. So now this is a whole thing. Why I have put it is because this is very Ascomycetis life cycle is very important for NEET. Okay, just check 1 second. Okay, so it is very important, very important for NEET. This is because this is small questions might come. I can tell you actually what kind of questions might come. I'll be telling you that. Okay, so this is very important for NEET. So now, second, okay, so I'll just explain what is happening in this feature. So as we know, Ascomycetis has, it's a life cycle. The life cycle has a sexual reproduction and sexual reproduction. Okay, so what happens is in the asexual reproduction, what happens? See, this is, suppose this is Ascomycetis. Okay, and as I said, they are totally dependent on spores and conidias. Okay, that is it. Conidias are these structures you can see. Okay, these are conidia, and from there, these conidias, they become mycelium. Okay, the base filaments, conidia together, they come together. Many conidias, they come together and found just make filaments out of it. Okay, this is just the asexual part. Okay, so conidia, okay, they fuse together to form mycelium. So once mycelium is formed, these filamentous structures will form. They'll eventually they'll grow into a fungus, as you know. Okay, so not very problematic. Now there is the sexual part which happens. So now this whole structure is divided into two kinds. Okay, there is the antheridium. Okay, this is the male part, and there is the ascogonium. This is the female part. This is the male part and the female part. Okay, so together what happens? 1 second. Together what happens? There's the male part and the female part. They come together. Okay, and they just fuse together. Okay, their protoplasms fuse together, not their nucleus. Okay, just their protoplasms fuse together, and this process is called plasmogamy. So these are haploids, one set of chromosome here, one set of chromosome there. They fuse together. They became chromosome 1, chromosome 2. They did not fuse. It's not 2n. It's n plus n. Okay, so they become ascogonium. Okay, so to sum it up, antheridium, as you can see, antheridium, okay, and then ascogonium. This is the male, this is the female. They fuse together, okay, in plasmogamy. This is haploid n and n. This becomes n plus n. Only the fusing of the cytoplasm. Okay. So, second. And then what happens? Just forms. Then there is the karyogamy. Okay. Then comes karyogamy. gamete, where the nucleus fuses. So what happens? N plus N becomes 2N. Okay, see, the nucleus fuses and they become N plus N. They become a diploid nucleus. So two sets of chromosomes. Okay. Then again they form their whole structure, the whole sac-like structures and everything, and they give out spores. Finally, formation of body with spores. Okay, I hope you understood. Let me just recap. Okay, so first there is asexual reproduction, just conidia. Together they come and they form a mycelium. Now the whole part comes is there is a sexual reproduction, where first there happens plasmogamy, the antheridium and ascogonium, the male part and the female part, they come together and there is plasmogamy, becomes 2 haploid, and then there is the whole sexual part where the nucleus fuses and it becomes karyogamy. The whole body forms and they start giving out spores. Okay, I hope it's clear for you. Okay, so there are no questions. We'll move on. This is the third part, the Basidiomycetes. Basidiomycetes also has a little bit of the reproduction thing. Basidiomycetes, as you know, they are parasites, rust, smuts and everything. Okay, the mycelium is branch and septate. There it was aseptate. This was septate, like this. Okay, we'll have divisions with nucleus. Asexual spores are generally not found, but vegetative reproduction is common. Okay, sex organs are absent, but plasmogamy, as I said, there's the main antheridium and ascogonium meets, that happens here. Okay, now they are the basic things what we eat: mushrooms, puffballs, etc. This is a type, they are type of Basidiomycetes. Okay, so as I said, not a lot of sexual reproduction and not a lot of things happening here. They have basically vegetative and kind of sexual reproduction. So there's basidiospores, okay, which are haploid in nature, and the antheridium and the ascogonium meet. Okay, the male type and the female type, they meet, and as I said, there is plasmogamy. They fuse together, okay, and they form the basidia. Now they form the zygote, they together. Then first there is plasmogamy, and then there is karyogamy, just the nucleus, different kinds of nucleus fuses together. Okay, there is cell division, and then there is the basidiospores. Okay, cell division happens, and then again spores are formed. To go again, they travel and they reproduce somewhere else. Okay, very simple, basidiospores. Okay, and then there's no asexual, sexual, there's no difference. Okay, basidiospores taken in, they mate, they form, and then they give out the spores. That's all. That's all you have to remember. This is also important for NEET. Please remember, I'm writing it. This is important for NEET. Okay, and the last one is Deuteromycetes. Deuteromycetes is actually, they do not follow regular reproduction cycle. Okay, they are not real ones. They are actually considered the litter form. Okay, because not a lot of ideas are actually known about it. That is why it's known as like that. Okay, asexual reproduction occurs by conidia, etc. Okay, and alternaria, celotrichum, trichoderma, and these things are belong to Deuteromycetes. Okay, now we are done with the plant, the Monera, Protista, and fungi. Okay, so now what? Okay, we might jump into Animalia and Plantae, but that is for the next sessions. Okay, one session I'll handle the Plantae. I might take 1 or 2 sessions, and then there is Animalia. Okay, so before that, before going, Okay, I'd like to talk about what R.H. Whitaker, the main man for these 5 sections, he forgot to put virus and viroids into the thing. Because this may be because virus outside host is actually non-living. So that is why he did not include it in the classification. So we'll just try to understand what they are. Okay, there is prions. Prions are basically protein particles. Okay, as you see, they are basically protein particles and they cause diseases. Okay, these protein particles might get inside animals. Okay, they might cause crepey in sheep, kuru in Malaysian tribes, CG disease. Okay, bovine spongiform. They'll go into the brain and they'll try to eat it up. Okay, mad cow disease. This is actually known as mad cow disease, and the cow will actually start rubbing their head against the bark of the tree anywhere, and eventually they'll die. Okay, so this is caused by prions. The next one, viroids. Those were prions were protein. Viroids are basically RNA particles. Okay, so RNA particles that cause diseases. Similar there they cause protein causing diseases. These are RNA protein causing diseases. Okay, they are basically go and affect the plants like potato spindle tuber, Citroexocortosis, cucumber pale fruit, tomato bunchy top, and all. These are some of the diseases. Okay. Viruses. We now know a lot about viruses because we are under the threat of coronavirus. Okay, this is the coronavirus is a type of virus. Okay, they are infectious, poisonous, have DNA or RNA as their genetic material. They're parasites. Outside, they are nothing. Okay, outside they are not living, but when they go into a host, they become living thing. And they were discovered by a person named Ivanovsky in 1892. Okay. And then there are lichens. Lichens are basically like they live on the host. Okay, like they'll also be giving some nutrition and they'll be taking some nutrition. It'll be a symbiotic relationship. Okay, and algae component is called phycobiont and fungal component is known as mycobiont. Okay, so this is the phycobiont and this is the mycobiont, and they are actually good pollution indicators. They actually help in indicating sulfur dioxide. Okay, so that is it. So now, any doubts? We are done today. We'll have some questions to discuss, of course. Okay, I have some just little questions to discuss, and then we wrap it up today. So if there are no doubts, we'll start with the question and answer. I'll give some time if anybody wants to answer. Otherwise, I'll give the answer by myself. Okay, so, okay, we'll start. The basis of classification according to Whitaker is cell structure, B, mode of reproduction, C, phylogenetic relationship, D, all of this. Anybody? Will anybody answer? Okay, so the answer to this question is basis of classification according to Whitaker, cell structure, mode of reproduction, phylogenetic relationship. As I said, Whitaker took everything. Okay, Whitaker took everything into consideration. So the answer is all of these. Okay, so the answer is all of these. Next, which of the following organisms were never included in Protista? Bacteria, red algae, slime molds, mosses. Anybody? Anybody wants to answer? Anybody? Okay, so the answer is mosses. As I said, everything. Okay, when Ernst Haeckel Okay, he said there is Protista, there is Animalia, and there is Plantae. He put everything in Protista. Okay, bacteria, algae, molds, and everything. But what he did not put was moss. Okay, moss was never considered as Protista. It always went into Plantae. Okay, so next question. Which characteristic place the fungi in a separate kingdom? I have discussed this just now. What? A, cell wall composition, B, cell wall structure, C, nutrition, D, nuclear membrane. I don't think anybody is answering. So the answer is cell wall composition, because it has chitin, which is not there in any other organism. So because of that, it is put into a separate kingdom. Okay, because of chitin. Remember this. This is a very important question for NEET. Okay. Next, which of the following is found in harsh habitat? Very easy, very easy. A, Eubacteria, B, Cyanobacteria, D, Archaeobacteria, C, Archaeobacteria, D, none of this. Okay. So the answer to it is Archaeobacteria. As I said, harshest of conditions. Archaeobacteria lives in harsh conditions. Eubacteria does not live in harsh conditions. Cyanobacteria definitely not, because it needs photosynthesis. So Eubacteria not a good answer. Cyanobacteria no. Archaeobacteria is the right answer, not none of this. Okay. Next, what is true of Euglena? A, presence of cellulose wall, B, presence of proteinaceous pellicle, C, presence of chlorophyll, D, presence of flagellum. Okay, so the correct answer here is presence of proteinaceous pellicle. Okay, as I said, Euglena, Euglenoids, ciliates, okay, protozoas, they have a thing called pellicle, a protein rich membrane. So the answer is presence of proteinaceous pellicle. Okay, that is the special thing. Next, longitudinal binary fission is found in A, Euglena, B, Plasmodium, C, Planaria, D, Paramecium. Anybody? Anybody? Okay, so I'll answer it. It is no, A, Euglena. Okay, this has the binary fission. Plasmodium, Planaria, Paramecium may have other kind of asexual reproduction, but longitudinal binary fission is found in Euglena. Okay. Protists obtain food as holotrophs, photosynthesizers, chemosynthesizers. Now, protist has a lot of things, as you know. It has chrysophytes, it has dinoflagellates, then amoeboids and everything, protozoas. Okay, so many things are there in protists, and some of them are photosynthetic, some of them are chemosynthetic, some of them are holotrophs. Holotrophs as in internal ingestion. Okay, okay, so these everything can be seen in protists. Okay, So the answer is D. Photosynthesizers, symbionts, and holotrophs. Okay. Next, a protein-rich layer which makes the body of euglenoids flexible is called, I have told this again and again. Okay, the answer is pellicle. Okay. Next, during unfavorable conditions, the plasmodium differentiates into fruiting bodies bearing spores at their tips. The group is Chrysophytes, dinoflagellates, slime molds, protozoans. Anybody? Okay, so the answer is, as I said, slime molds. Okay, as I said, sacs-like structure. Okay, there'll be one different places. Suppose there's no food. Okay, they'll merge together and become a big sac-like structure. Okay, these are slime molds. So the answer is slime molds. Okay. Next, the member of which following are litter decomposers? I said it just now. A. Deuteromycetes, Ascomycetis, Basidiomycetis, Phycomycitis. The answer is Deuteromycetes because no proper idea about its reproduction. That's why it's called as litter decomposers. Okay. So, last question. Lichens are very good pollution indicator because they are sensitive to, I said it just now. The answer is sulfur dioxide. Okay. SO2. Lichens have a very good tendency to identify SO2. So the answer is SO2. Okay, so the end of the class. I hope you have no doubts. If you have any doubts, you can log on to the Practically app and you can ask your doubts also there. You can subscribe to the app and you can get all kinds of courses. Okay, you can get the interactive videos, everything, and you can also rate your experience over there from your phones. Okay, so, and this was just the biological classification for the basic three kingdoms, Monera, Protista, and this thing. So the next one will be about, Monera, Protista, and fungi, and the next one will be about Plantae and Animalia. Okay, so I hope you follow us there. Okay, you follow us on YouTube, you like your experience and share it with your friends. Okay, I'll see you later. Okay, bye bye. Have a very good day.